Okay, so welcome back to this next video in which we are discussing G protein coupled receptors. Okay, so in the previous video what we discussed is a very basic model for what a heterotrimeric G protein looks like. Okay, so here's a very basic picture of a heterotrimeric G protein consisting of an alpha, a beta, and a gamma subunit. Okay, and we discussed how heterotrimeric G proteins can be in two states. The off state, in which the whole thing is as a trimer and the alpha subunit has guanosine diphosphate bound to it, and it's this that we've drawn here. And then what's going to happen is when this comes into contact with an activated G protein coupled receptor, the G protein coupled receptor can catalyze the conversion of this heterotrimeric G protein from the off state to the on state. And in the on state, what's going to happen is the alpha subunit is going to end up with GTP bound to it, and that's going to change its conformation so that it can no longer bind to the beta-gamma complex. Okay, and the two are going to separate off, and we want to look at the actual molecular detail of what actually is involved in that. Okay, so we then in that previous video looked at all the different alpha, all the different beta, and all the different gamma subunits of heterotrimeric G proteins. Okay, right. So firstly, in this video, what we want to do is go to a slightly more advanced picture of what a heterotrimeric G protein looks like. Okay, and this is the picture that's going to be useful for us in understanding the activation of heterotrimeric G proteins. Then what we're actually going to do is look at the full-on structure of an alpha subunit of a heterotrimeric G protein so that we can understand how, when a heterotrimeric G protein binds to the G protein coupled receptor, how that actually causes the release of the guanosine diphosphate from the alpha subunit of the heterotrimeric G protein. Okay, right, so let's now do a fancier model of what a heterotrimeric G protein looks like. Okay, so we'll start off with the alpha subunit of a heterotrimeric G protein. So the first thing to say is that alpha subunits can be divided up into two main domains. Okay, a domain known as the GTPase domain, which is this first domain that I'm drawing up here. Okay, so this big domain here, this is the GTPase domain, okay, and then a smaller domain which is below here, which is known as the helical domain. Okay, so this portion is called the helical domain. Okay, right. Now, this little site that I have left here, this is going to be the site for binding to guanine nucleotides, so binding to GDP and GTP. Okay, and initially, when the heterotrimeric G protein is in the off state, you're going to have GDP bound there. Okay, so the binding site for the GDP is within the GTPase domain of the alpha subunit of the heterotrimeric G protein. Okay, so let me just explain why these different domains are called these different things. Okay, so the GTPase domain, that's called the GTPase domain because it is a GTPase enzyme. It has the capacity to hydrolyze GTP to GDP, and we'll discuss how important that is later on. Okay, the helical domain is called the helical domain because it has a huge number of alpha helices making it up. Again, which we'll see when we look at the alpha subunit of heterotrimeric G proteins in more detail. We'll see the alpha helices which actually make this helical domain up here. Okay, now, the GTPase domain and the helical domain are linked by two linkers. Okay, so here, this first linker that I'm showing there, that will be linker 1. Okay, and then here, this is a linker 2 here, and then there's a special region here that I will draw, which is known as switch region 1. Okay, and this is going to be very important for the alpha subunit of the heterotrimeric G protein to bind to the beta subunit. So I'm going to colour this in. So after that little linker there, L2, okay, you then have this very, very important region, and I don't want to label it right there because I want to draw the beta subunit there in a moment, which is known as the switch region 1. Okay, and you can't read anything about heterotrimeric G proteins in the literature without knowing what the switch regions are. So here's the first switch region of the alpha subunit of the heterotrimeric G protein, switch 1 there. Okay, and as I say, it's going to be important in binding to the beta subunit. Okay, now let me just add another one of these switch regions on, which is here, which is switch region 2. So that's switch region 2, and I'm abbreviating switch down to just SW, so this stands for switch. Okay, so switch region 1 and switch region 2, and these are both in the inactive state going to be extremely important in binding to the beta subunit of the heterotrimeric G protein. And in fact, when you bring in a GTP there rather than a GDP, what's going to happen is that the switch regions are going to bend in towards the GTP, and that's going to interfere with the interaction between the alpha and then the beta subunit, and that's why the two dissociate apart. Okay, so these are going to be very, very important, switch region 1 and switch region 2. Okay, right. To add on some more very important domains here, okay, we'll firstly add on the N-terminal alpha helix, which I'll put here. Okay, so here's the amino terminus of our alpha subunit of our heterotrimeric G protein. And at the amino terminal end, you have an alpha helix here, which I'll colour in orange, okay, known as the N-terminal alpha helix, which for short is just abbreviated to alpha N. Okay, now I should also just mention that, remember when we were talking about the simpler model of heterotrimeric G proteins, we talked about how the alpha and the gamma subunits have lipid moieties added onto them. The alpha subunits specifically have palmitoyl or myristoyl groups added onto them. They generally get added on prior to the alpha N helix here. Okay, so this will be that lipid moiety there. Okay, so I might even draw the plasma membrane there with that lipid moiety sticking into it. Okay, brilliant. Right, then to put on some more domains that we're going to need later on. Okay, so I'm just in this picture marking on the really important domains. In a moment we're going to look at the entire structure of the alpha subunit, and we're going to see how all of these domains fit together in the whole thing. Okay, but for this picture, I'm just marking on the really, really important domains, okay? And these domains in orange, the switch region 1, the switch region 2, and the alpha N, they are going to be important for binding to the beta subunit, which I'll draw on in a moment, okay? The domains that I'm now going to colour in vivid purple, these are going to be the domains that are going to be really important in binding to the G protein coupled receptor once it's in the activated state. So there is a little bit here after the alpha N helix here, okay? And this is known as the alpha N beta 1 linker, okay, because it's between the alpha N helix and the beta 1 beta strand, and we'll see the beta 1 beta strand when we look at the entire structure of the alpha subunit. Okay, that will be in here in this bit that we've just abbreviated down to a great big blob. Okay, right, then the other portions that are really, really important for binding to the to the G protein coupled receptor are the C-terminal portion of the alpha subunit, which is here. So here's the C-terminal, and then that portion prior to the C-terminal there in pink, that's really important for binding to the G protein coupled receptor. Then you have some more loops extending from the top of the alpha subunit like so, okay, and these loops are called the alpha 4 beta 4 loop. Okay, that's this one here, between the 4th alpha helix and the 4th beta pleated sheet. Oh, sorry, not the 4th beta pleated sheet, let me just correct that. It's the 6th beta pleated sheet, as we'll see later on. Okay, and then we have another one of these loops here between the 3rd alpha helix and the 5th beta sheet, okay? As I say, when we draw out the full structure of the alpha subunit, you will see how these all fit together and how they are between alpha helices and beta sheets. So this is alpha 3, beta 5 here. Okay, so there's going to be an alpha helix here called alpha 3, there's going to be a beta sheet here called beta 5, then there'll be an alpha helix called alpha 4, then a beta 6 here, and then actually there'll be an alpha 5 as well. Okay, so we'll see that once we actually look at the full structure of the alpha subunit of the heterotrimeric G protein. Okay, right, so that's my more sophisticated picture of the alpha subunit of the heterotrimeric G protein. It has the really important bits marked on here, but it doesn't actually show the full structure, and this is the picture that we're mainly going to use to understand the activation of these heterotrimeric G proteins. We're only going to go to the full structure of the alpha subunit so that we can understand what actually triggers the GDP release, okay, because to see that we really need to know a lot about the structure of the alpha subunit. Okay, right, so there's our alpha subunit. Okay, now let's add on to this picture the beta and the gamma subunit. So we'll start with the beta subunit. So the beta subunit has a very important structure that is key to it, which is known as the beta propeller. Okay, so it has a beta propeller structure. Now, let me explain what a beta propeller is. Basically, it's called a beta propeller because it looks like a propeller that you might have at the end of a ship, for instance, that powers a ship forward. Okay, and basically the blades of the propeller are going to be made up out of beta sheets, which is why it's called a beta propeller. Now, the beta propeller that's going to be present in the beta subunit of heterotrimeric G proteins is what's known as a 7-bladed beta propeller. Okay, and that means that it's effectively going to have 7 blades, so 7 blades that are produced out of beta sheets. So let me mark these on. So I'll put one of these beta propellers here. Okay, and there is only one in the beta subunit of the heterotrimeric G protein. So here we are, 1, 2, 3, 4, 5. So we now need 2 more, like so, and I'll mark it in like so. Okay, so here's one of those blades, which will be made up out of beta strands, all going parallel to one another to make that blade, and then you'll have another one here, which will have beta strands parallel to one another to make that blade, and then you'll have another 5 here to make the 7-bladed beta propeller here. Okay, so this is the absolutely key structure of the beta subunit. This is the dominant structure of it. Okay, and that alpha N helix here, that will be binding to the top of the beta propeller here, which will then have its C-terminus coming out here. And in fact, the C-terminus of the beta propeller, or well, rather of the beta subunit, is actually going to be involved in binding to the G protein coupled receptor. So I'll just colour that in vivid purple, like all the other portions that are going to bind to the G protein coupled receptor have been coloured in. Okay, right. Now, the beta subunit of heterotrimeric G proteins does consist of more, however, than just this 7-bladed beta propeller. It also has an amino terminal end, which has an alpha helix. Okay, so let me just show this. So coming down from here, you then have an alpha helix here, and then the N-terminal portions down here. Okay, right, so I'll colour this alpha helix in, I think, in green. So here's the alpha helix of the beta subunit of the heterotrimeric G protein. So those are the major features of the beta subunit of the heterotrimeric G protein. You have the C-terminus here, which will bind to the alpha N helix of the alpha subunit, and also which is involved in binding to the G protein coupled receptor. You then have the beta propeller, which is going to be binding to these switch regions here, switch 1 and switch 2. Okay, and then down here you have this alpha helix here towards the amino terminal end, and this is going to be important in binding to the gamma subunit. Okay, right, so to stress again, where are the major contact sites between the alpha subunit of the heterotrimeric G protein and the beta subunit of the heterotrimeric G protein? It's these switch regions, switch 1 and switch 2 here, and also the alpha N helix here. These are the major contacts between the alpha subunit and the beta subunit of the heterotrimeric G protein. Okay, right, yeah, let's now show the contact between the beta subunit and the gamma subunit. So let's put on the final subunit here, which is the gamma subunit. Okay, so basically the gamma subunit also has an alpha helix that is at its N-terminal end, and what actually happens is the alpha helix of the gamma subunit and the alpha helix of the beta subunit form what's known as a coiled coil domain. Okay, so the two alpha helices coil around each other to produce a coiled coil domain. So let me just explain this in a little bit more detail. So basically, we know that an alpha helix is this sort of structure here. It's a helix, and the alpha, remember, means that there's approximately 3 amino acids on each term. Okay, now to make a coiled domain, what you're going to do is basically you're going to coil the polypeptide in this alpha helical structure around another polypeptide that's also in an alpha helical structure. So like so, here is one of these alpha helices, and then the other alpha helice is going to coil and intertwine around this, like so. Okay, and this is how you produce one of these coiled domains. Okay, by coiling alpha helices around one another. So because an alpha helix is already a coil, okay, just looking at a single alpha helix, that's already a coil, and we're now coiling coils, that's why it's called a coil domain. Okay, so this is going to intertwine the beta and the gamma subunit together down here. Okay, then the gamma subunit will continue on up here. Okay, like so. Its C-terminus is actually also involved in binding to the G protein coupled receptor, so I will push on its C-terminus here. Okay, and it will also have a lipid moiety attached to its C-terminus here, that farnesyl group, or that geranyl group. Okay, that we mentioned right at the start when we're looking at these simple pictures. Okay, so here's the lipid moiety attached to the gamma subunit, here's the lipid moiety attached to the alpha subunit. Okay, right. So that's our more advanced picture of a heterotrimeric G protein. What we now want to look at is we want to look at the actual structure of an alpha subunit of a heterotrimeric G protein. And the reason we're going to do this is because we want to understand when the heterotrimeric G protein binds to the G protein coupled receptor, which we haven't done yet. This is just the inactive heterotrimeric G protein with GDP bound to it. We want to understand when the heterotrimeric G protein binds to the G protein coupled receptor, what conformational changes are going to occur in the alpha subunit that allows the release of the GDP. Okay, and we don't have enough information with this picture to understand that yet. And really, to understand that fully, we need to actually look at the entire structure of the heterotrimeric G protein. Okay, so I'm going to go over onto a fresh sheet of paper to start this picture. Okay, so this is a big structure. This is not trivial, what we're about to do. We are going to look at the complete structure of an alpha subunit of a heterotrimeric G protein. Now, to orient you as to what sort of an angle we're going to be looking at it, really, we're going to be looking at it at this sort of an angle here, okay? Looking at it from the perspective of the beta-gamma subunit here. Okay, so we're going to have these switch regions at the front, and then towards the back we're then going to have these loops that are involved in binding. Okay, right, so let's have a look then at the alpha subunit of a heterotrimeric G protein. So we'll start with what we've seen in the previous picture, which is the amino-terminal end of the heterotrimeric G protein. Okay, so here, the alpha subunit of the heterotrimeric G protein. So here's the amino-terminal end here. Okay, and we previously coloured that alpha N helix in orange. I'll stick to that. Okay, so here we go. Okay, like so. So that's the alpha N helix here. Okay, then remember what we had after the alpha N helix. After the alpha N helix, we had that little portion that was going to be involved in binding to the G protein coupled receptor. And I'll mark that on again. Remember, that was called the alpha N beta 1 portion because it linked the alpha N helix with the beta 1 strand. And now we're going to actually get to see the beta 1 strand. So let's now mark on the beta 1 strand. So here comes the beta 1 strand, and then it will curve around like so. Okay, right, so let's mark on the beta 1 strand. So I'm going to show a beta strand in turquoise, and I'm also going to put an arrow for which direction it's going in. So that's our first beta strand of this structure, and I'll label that up here. That's the beta 1 strand there. And I'm making these labels deliberately very close because I know we're going to have lots of other things around here later on. Okay, so that's why I'm trying to keep the labels small and tight in. Okay, right. Then after the beta 1 strand, you have a portion that's actually going to be really important in producing the binding site for the guanosine nucleotides, okay? So this portion in red. So I'm going to cover all the portions that are involved in binding to the GDP or the GTP in red here, as we did in this picture here. Okay, so this is one of these important portions for making that binding site for the GDP or the GTP. Okay, and this is what's known as the P loop here. Okay, right. Then after the P loop, it's going to curve along like so, and then we're going to go down and produce the helical domain. Okay, so I'll curve down like here. Okay, and then we'll draw the helical domain in just a moment. Okay, right, but before we go into the helical domain, in yellow here we're going to have an alpha helix that's not part of the helical domain. I'll colour all the alpha helices that are going to be part of the helical domain in blue, as we did here. Okay, this is actually what's known as our first alpha helix of the GTPase domain. Okay, and this will be called alpha 1. Okay, the first alpha helix of the GTPase domain. Okay, then you have this linker that takes us down into the helical domain. Okay, and this is linker number 1. Okay, and then one of the major portions of the helical domain is a very large first alpha helix here, and I'm going to colour this alpha helix in blue because all of the alpha helices of the helical domain are going to be coloured in blue. Okay, so here's a very large alpha helix of the helical domain, and this is known as alpha A alpha helix. So all of the alpha helices of the helical domain will be called alpha for alpha helix, and then you'll have a letter denoting their order basically in the helical domain. So this is the first helix of the helical domain, so it's alpha helix number A, or letter A rather. Okay, then what we have is another alpha helix coming down here. Okay, and this will be the second alpha helix of the helical domain, and this will be called alpha helix B. Okay, then it will loop back, it will loop under alpha helix A here, so it's looping under alpha helix A, and it will go right back over to here. Okay, and here's the third alpha helix here. Okay, and this is what's going to be known as alpha helix C. Okay, then it will loop up like so, loop round like so again. Okay, and here what we're going to have is a little alpha helix called alpha helix D. Okay, so this is called alpha helix D. And then coming down here, you've got the fifth alpha helix of the helical domain, which is called alpha helix E. Now I just want to point your attention to a portion that's going to become important later on. Okay, this section between alpha D and alpha E here, this is known as the alpha D alpha E loop. Okay, and that's going to become important later on. Switch region 3 is actually going to be contacting the alpha D alpha E loop. Okay, right, so as I say, we'll come back to that later on. Right, so all of this is still the helical domain. All of these blue alpha helices are all the helical domain. Okay, we're going to have one more alpha helix here. Okay, which is alpha F, which I'll colour in again blue here. Okay, and now we're finally going to leave the helical domain. Okay, so here's link number 2 here. Okay, and then you're going to have an important region that we've seen before, which is switch region number 1. Okay, so I might colour that in orange again. So here, this is switch region number 1, that really important region. If there are regions of the alpha subunit or the heterotrimeric G protein that you need to learn, they're the switch regions. They're the important regions. Okay, so here's switch 1 just after linker 1. Okay, and now we're out of the helical domain, so I might highlight up all of this down here is that helical domain that we saw in the cruder picture overneath. Okay, so this is the helical domain of the alpha subunit of a heterotrimeric G protein. Okay, right. So what's going to happen after switch 1? Well, we're going to have more beta strands with E, okay, which are going to form a nice little beta sheet with beta 1 here. Okay, so here comes some more beta strands. This is beta strand number 2 here. and then we've got beta strand number 3, and they're both sitting nicely parallel with beta strand number 1 to form a beta sheet here. So this is beta 2, this is beta 3. Okay, then after beta 3 you have another really important domain that we've seen before, switch region number 2. Okay, and again look at how close these are to this place where you have this GDP or GTP binding site, which is this P loop here. And I should have just said switch region number 1 is going to go over that alpha 1 helix here. It's going over the top of it. So this is in front of alpha helix number 1 here in yellow. Okay, and again the switch region number 2 that I'm about to show here, this is going to go over that P loop that we've got there. Okay, in red. So here now is switch region number 2, and you can see how close they are to where that binding site for the GDP or the GTP is going to be. Okay, right, so that's switch region number 2. I don't know if I want to put the label there, I'm going to leave that space, but that is switch region number 2 there in orange, so remember that that's switch region number 2. Okay, then after switch region number 2, what you're going to have is the second alpha helix of the GTPase domain. Okay, and this is going to be going up like so. So here's the second alpha helix of the GTPase domain, and it's going to fold all the way around like this, and then you're going to have another beta strand, which will be in parallel with the rest of them there. Okay, so let's colour this in. So here is our second alpha helix of the GTPase domain, which again I'll show in yellow. So this is called alpha 2. Okay, and then we have our fourth beta strand of the GTPase domain, again sitting in this lovely beta sheet that we're forming here. So this is beta 4 here. Then what will happen is the polypeptide will go under switch region number 2 here. So again, this is underneath switch region number 2. And then we'll fold back round like so, and then go up. Okay, and we've got another important domain right down here in orange, which is switch region number 3. And switch region number 3 is going to be contacting the alpha D, alpha E loop. We didn't show switch region number 3 on that previous picture, and it's because switch region number 3 isn't as important as switch region 1 and 2 in binding to the beta subunit. In the previous picture we showed the ones that were really important in binding to the beta subunit. Switch region number 3 is important in connecting the GTPase domain with the helical domain below. Okay, so this is the interaction here between switch region 3 and alpha D, alpha E here. Okay, then after switch region number 3, you then have your third alpha helix of the GTPase domain here in yellow. And now I'm going to go on to showing this in a separate picture, okay, because what's going to happen now is this thing is going to fold back round and round behind all of what we've drawn now. So I'm going to draw another picture here, which is the continuation of alpha 3, but it's all behind what we've shown so far. Okay, right, so to continue this on, I'll redraw the alpha 3 helix here. Okay, so this is this exact same helix continued on here, just drawn in a separate picture because it's difficult to show it all in one picture. Okay, so I'm just now continuing it on, and it's all sitting behind what we've shown so far. Okay, and then what's going to happen is basically you fold back round like so. Okay, and here is that first really important loop that we saw on the more crude structure. Okay, this is going to be the alpha 3 beta 5 loop. Oops, missed out the beta. Beta 5 loop. So let me try and connect these two. Okay, so this loop here, alpha 3 beta 5 that we saw over here, which is really important in binding to the G protein coupled receptor. And of course that means that we must have a beta sheet here, which must be beta 5. Okay, and indeed we do. So this is beta 5 here. Okay, then it's going to loop around again. So you have a little loop here, and then you loop back up Okay, like so, and then back around. Now, at the bottom here, we actually have an alpha helix that extends into the helical domain, another alpha helix of the helical domain. Okay, so this is the portion of the helical domain that isn't connected with all the other portions as far as the primary structure is concerned, but in the tertiary structure ends up being connected back with the helical domain here. And this is the 7th alpha helix of the helical domain, alpha G. So if you can imagine positioning this correctly, this would sort of be around down here with the other alpha helices of the helical domain. Okay, right. Then after that alpha G alpha helix, we have the 4th alpha helix of the GTPase domain here, okay, which is known as alpha 4. Okay, then of course we have another important loop here, which is the alpha 4 beta 6 loop here. Okay, so this is called alpha 4 beta 6. Okay, then it comes back down like so, with obviously the beta 6 beta strand here. Okay, so here's another beta strand like so. This is beta 6. And then finally, you have a really important loop here known as alpha 5 beta 6 loop, okay, which is another portion that makes up part of the GDP-GTP binding site, the guanine nucleotide binding site there. So I'll colour this in red, okay, and this has a really important motif in it known as the TCAT motif. Okay, again, to understand that you need to be able to read single letter amino acid code. T is for threonine, C is for cysteine, A is for alanine, and then T obviously is for threonine again. So you have this sequence of 4 amino acids always in this alpha 5 beta 6 loop, and that's really important in making the other part of this guanine nucleotide binding domain here. Okay, so the P loop and this portion together are positioned together to make this little binding pocket for GDP and GTP there. Okay, then after alpha 5 beta 6, what you then have is the alpha 5 alpha helix, then ending with the C-terminal up there. Okay, so here in yellow, that's the alpha 5 helix here. So that's alpha 5. And then after the alpha 5 helix, what you'll then have is that C-terminal domain, which is important in binding to the G protein coupled receptor. And then we have finished. There you go. Something you might never have thought you would ever see. There is the actual structure of an alpha subunit of a heterotrimeric G protein. Beta sheet and alpha helix by beta sheet and alpha helix. Okay, right. Now you might think, well, why on earth do I need to know that? Basically, if we're going to understand the actual conformational changes that occur in the alpha subunit that allow the release of GDP when it binds to the G protein coupled receptor, we need to know this. That's the only reason we need to know this, okay, to understand the conformational changes that lead to the release of the guanosine diphosphate when the G protein binds to the G protein coupled receptor. Okay, right, so we're going to abandon this for now, okay, and we're going to go back to our cruder picture up here, okay, and we're going to have a look at how the heterotrimeric G protein binds to the G protein coupled receptor, okay, and then we'll come back to this picture after that to look at, once it's bound to the G protein coupled receptor, what conformational changes are going to occur that lead to the release of the GDP. Okay, so in the next video what we'll do is firstly look at how the heterotrimeric G protein binds to the activated G protein coupled receptor. We'll say something about specificity, and then we'll move on to what conformational changes actually lead to the release of GDP, and then we'll talk about GTP binding and how that's going to lead to the dissociation of first the beta gamma subunit and then the alpha subunit from the G protein coupled receptor, and then it leads to the signaling